What I want to do with Chapter 1 today is give you an appreciation for why you're studying microbiology, okay, and how this affects you. We're not going to talk about everything that's in the chapter. Of course, I don't want you to memorize everything out of the chapter. What I would test you on would be the things that are written on the PowerPoint slides. But I'm going to kind of pick up on a few other things as we go through in our little discussion, okay? So the first thing we need to understand if we're going to take a microbiology class is, okay, microbiology is the study of microorganisms. So what is a microorganism? A microorganism or a microbe is anything that is too small to be seen without a microscope, okay? And that's really still not as good of a definition as we could get. A better definition, we need to add the word living into that, okay? So we're going to talk about everything that is alive that we can't see with our naked eye. That's what we're going to cover in this class. So you can already see that's going to be a lot of stuff, right? I mean, what do you think is in this room right now that you can't see? If you ever sat down and just thought about it, do you think there's a lot of microbes in this room right now? Yes. As I'm talking, you can't see it, and this will sound gross, but there is stuff erupting out of my mouth with every breath. Every time I talk, minute pieces of moisture are coming out of my mouth that are filled with bacteria. And you may all think, well, that's just disgusting. But every time you breathe, things are coming out of your mouth. Things are coming out of your nose. Okay? In reality, we may not want to think about it, but there are probably things coming out of other places in some of our bodies in this room as we sit in here right now too. And that's just what we're putting in the room. There were already tons of bacteria, tons of different fungi, all kind of stuff in this room. Okay? We're going to learn what all those little things are this semester. Okay? Now, as I talk about it, it automatically starts sounding, again, like we're just going to talk about the bad stuff. Okay? So what's written on this slide are some of the good things that microorganisms do for us. Okay? Number one most important thing microorganisms do for us is they form the basis of every food chain on Earth. They form the basis of the food chain in the water as well as on land, your terrestrial organisms. Okay, we all know what the food chain is, right? If you don't have the bottom of the food chain, when you get all the way up to the top of the food chain, which is us, we would have nothing to eat. Okay? So y'all don't seem very impressed with that. Give you something else. Do you guys enjoy breathing oxygen? Yes. Okay. Where do you think you get oxygen from? There's no wrong answer. Trees, plants. That's what you usually learn, right? Only about 20% of Earth's oxygen comes from plants and trees. So guess where the other 80% comes from? Algae. Algae is a microorganism. So saying that another way that makes it sound a little more impressive, you could cut down every plant and every tree on Earth, and we would still be able to breathe perfectly fine because we have algae. Okay? So that's a pretty good use for a micro, right? It produces oxygen for us to breathe. Okay? Well, if some of you want to argue and say, well, we still need plants and trees for other things, plants and trees need water to grow, right? They use their roots to collect water. Guess what helps them do that? Fungi. There are microbes in the soil that help the roots expand their surface area so they can get more water so they can grow. Okay? So that's pretty helpful, right? How do they help us directly? Inside of your intestines, you have tons of bacteria. Why are they there? Well, number one, they keep bad bacteria from being able to grow. They also help produce vitamins. Okay? Some vitamins we would never have if we didn't have bacteria living in our gut to produce them. Other vitamins, we wouldn't be able to absorb them, meaning use them in our body, if we didn't have the bacteria in our intestine. Okay? Things that we eat have microorganisms in it. Do you guys know of anything? What's something you eat or drink that has microorganisms in it? Yogurt, milk, very good. Milk actually has algae in it. That's what stabilizes milk and yogurt as well, so it can sit on the shelf longer. So if any of you want to say, I don't eat algae, that's gross, yes you do, if you drink milk. What else? What about liquor? Do any of you drink liquor? Okay, some of you are, yeah, I do. Okay, this conversation just got interesting. If you drink liquor, you eat yeast. Yeast is a fungus. That's actually what makes the liquor. Okay? If you don't drink liquor, if you're a good little boy or a good little girl, do you eat bread? Right? We all eat bread. The same yeast that makes wine is used to make bread. The ethanol just generally evaporates as we're cooking the bread. That's why bread doesn't taste like liquor. Okay? Tons of other things we could talk about. Just want you to have an overall appreciation. And throughout the semester, don't lose that idea. Bacteria and other microorganisms are not bad. Most of them are good. There are a few of them that will make you sick. And so the term we use for a microorganism that can make you sick is the term pathogenic. So you will hear that word a lot as we go through the semester. If it makes you sick, it's a pathogenic microorganism. All other microorganisms are perfectly fine. Most of them are very good for us. Okay? Now, of course, I didn't cover everything that was in this first chapter, but there's some really neat things in there. Our sewage treatment plants, we use microbes to treat sewage so that it can be turned back into water and we can reuse it. So if you want a gross thought for the day, what you flush down the toilet is eventually what you drink again. Kind of nice to think about, but it is. Okay. If you're wearing blue jeans are produced using microorganisms. So there's lots of really just cool things you can read about. Okay. All right. So a couple things we need to know from biology class. First off, how do you properly write the name of any organism? So this is micro class. So how do we write the proper name of a microorganism? They always will have two names. They have a genus and a species. Does that sound familiar to you guys? Okay. So if we were going to write our name, let me tear something up. We would say that we are Homo sapiens, right? Okay. Actually, if you want to look up something neat, we have a subspecies now because we have officially evolved. We're not just Homo sapiens, we're Homo sapiens something. I don't remember what it is, but it's a neat little article. Okay? So that's our name. We're Homo sapiens. So which word is the genus? Homo. So genus is first, species is second. The genus is always capitalized. The species is always lowercase. If this was being typed, it would need to be italicized. And you'll see that a lot as you read through your text and we start talking about these microorganisms. If I wanted to abbreviate this, what's the right way to abbreviate it? Very good. Yeah, they couldn't tell me yesterday. Very good. So it would be H. sapiens. Okay. That's perfectly fine. There's nothing wrong with abbreviating the genus, but you do not abbreviate the species. This will become very handy because not all things have a genus name that is as easy and short as Homo. Okay? That tapeworm I gave you the example of, Diphyllobothrum latum. Well, if you start taking a test and you start thinking, crap, I have no idea how to spell Diphyllobothrum, you just write a D, and then you write latum. Okay? So it's a very important tool, not only to make a good grade in a class, but if you're going to do anything in the scientific field, you learn to abbreviate that genus name. My MLT guys, that probably won't work when you actually get into your MLT classes, but it'll work for me. So you can get half of it from me, and then they'll torture you a little more when you get to them. Okay? All right, so that's our proper way to name things. Now, please don't try to memorize this stuff. This is just my way of showing you these are the groups of organisms we're going to talk about this semester. We're going to spend most of our time talking about bacteria, just because they are the most common microbe. Bacteria are your prokaryotic cells. Anybody know what a prokaryote is? Okay, we're eukaryotes. Good. Prokaryotes are bacteria. Well, what's different about us? All prokaryotes are unicellular, but some eukaryotes are too. Okay. If you don't know, it's not a big deal. This is chapter 4. We're going to review all of this. don't have a nucleus. That's the most common answer I get. That's actually not quite right. That's the way we teach it to you in biology class. The true difference is a prokaryote doesn't have any membrane-bound organelles. So some of your prokaryotes do have organelles. They're just not fancy, so they don't have a nucleus. They have mitochondria, and that's what we're going to cover in Chapter 4. You spent most of your time in your biology classes learning about everything but bacteria. So that's why we're going to spend most of our time talking about bacteria. But we're also going to spend a fair amount of time talking about microscopic fungi. So that's not mushrooms. That's a yeast or a mold. Okay? We all know what mold is, right? You ever seen mold on your bread? Okay? How many of you have ever just taken the moldy pieces out and left the non-molded pieces and ate them? Oh, don't act like you haven't. Okay, your parents did it to you, okay? But if any of it has mold you can see, all of it has the microscopic form at that point. But it's not hurt you. If mold was going to kill you, none of us would be here. I mean, if your parents didn't do it to you, I guarantee you your grandmother pinched the mold off and gave it to you. They grew up in the Depression. They know how to be thrifty. All right, I digress. We're going to talk about protozoans, really cool little single-celled eukaryotes that are almost like little animals, can make you really sick, really nasty diseases that are associated with those. We'll talk a little bit about algae. The last two groups we'll talk about, it's kind of weird for them to fit into microbiology class. We don't really have anywhere else to put them. Your parasitic worms, that's what helminths are, they're worms. They're huge. But the reason we study them in micro class is because when they are transmitted from one person to another, they're microscopic. So a tapeworm, for example, the record is 28 feet inside of a human's intestines. Well, I mean, that's obviously not microscopic, right? But whenever you get that tapeworm, you can't see it. Okay, so if you've ever thought you saw a worm in meat or something that you bought, it wasn't a worm, it was something else. Okay. The last group, viruses, they're not alive, so they're not technically microbes, but most of you have probably never spent any time talking about viruses, and I want you to know a little bit about them because you've all had a virus at some point. So it's good general knowledge. Okay? So you're going to keep this slide with you, and you're going to refer back to it as we go throughout the semester. This will help you a lot when you study for your final exam. Okay? Hint, hint. All right. So the last thing we're going to do today is just talk about a few different people and what they did that was important. Not a history teacher, obviously. Okay? I don't have to memorize dates. I don't know them. Okay? I just kind of have a general idea of what century things happened in. But all of these people that are in this slideshow did something important in the field of microbiology, and we're going to keep referring back to them as we go throughout the semester, so you need to know what they did. The first important person to discuss is Carl Woese. Okay? Carl Woese was the first one to figure out that we were classifying all of our organisms a little bit incorrect. Okay? Back when we first started classifying organisms, we put everything into 5 kingdoms, right? How many of y'all learned those 5 kingdoms? I did. Right? There's nothing bad about your education. That's just what was accepted. Well into the 80s and 90s, we just taught the 5 kingdoms. Well, back in 1978, Carl Woese said, guys, those 5 kingdoms aren't right. And he proposed that instead of having 5 kingdoms as the top tier, we should have domains and put everything into 3 domains. The domains would be the bacteria, the archaea, and the eukarya. And then the kingdoms, we need to have 4 kingdoms, and the kingdoms belong under the eukaryotes. Okay? He said this in 1978. We did not start doing this until the early 90s when we got a little bit more evidence, when we started doing DNA sequencing and things like that. But when we go back to it and we realize he was right, this is the proper way to classify things. Because if we put something in the domain bacteria, that means it's a bacteria that's got a normal cell wall. We're going to learn about the cell walls a little later. If we put it in the domain archaea, that's the super old bacteria. That's the ones that live in boiling lava inside of a volcano, like just really crazy microbes. Everything else is a eukaryotic cell. It evolved later, so it needs to be in the domain eukarya. Okay, so it's a really important distinction so that we can classify our microorganisms correctly. So that's what Carl Woese did. Okay. So this next slide, again, please don't try to memorize dates. But this is a list of kind of a timeline of what important people did to allow us to study microbiology. Okay? So the definition of a microbe, too small to be seen without a microscope, right? So what did we have to invent before we could study microbiology? Microscope. Okay? First guy to invent a microscope was Robert Hooke. But when you see his name in a science book or a history book, he's known as discovering the cell theory. Okay, well, how does that go together? What he did, he took some magnifying glasses and stacked them on top of each other, and then he took a piece of plant and looked at it under his microscope. When he looked at it, he noticed that the inside of the plant looked like this. And his thought was, you know, that kind of looks like gel cells, okay, connecting cells in a jailhouse, okay? And then he said, you know what? I bet everything that's alive is made of these little bitty compartments. And he just decided he was going to call those compartments cells. So that's where we get the word cell. All living things are composed of cells. So he's on his establishing the cell theory. Now that was in the 1600s. So that was actually pretty good for the 1600s. But he never saw any microorganism. He was looking at big things like a leaf and just looking at the cells of the leaf. He was looking at big eukaryotic cells. It was not until the 1700s that we first had proof there were things that were alive that were too small to be seen without a microscope. And that didn't happen until Anton von Leeuwenhoek. Okay, I don't know, he's German. This guy invented a real microscope. Okay? When he invented the real microscope, he was able to take pond water and see things that were microscopic wiggling around in the water. So he called them animalcules. They were actually microorganisms. We think they were protozoans, and we do think he even saw some bacteria. Okay? You notice nothing really substantial happened in our timeline from 1723 until the late 1700s, early 1800s. So why did nothing happen for 80 years? Because while Leeuwenhoek was alive, he would let people come look through his microscope and see his stuff. But every time he would leave the lab, he would take parts off of his microscope. He didn't want anybody else to know how to make it work. And it was not until he died and several years later that we finally figured out how to rebuild his microscope. So just kind of a, I don't know, kind of an interesting story for a science nerd that he was so proud of his invention he didn't want anybody else to have it. So he took parts off. So it took us quite a while to be able to do it again. But eventually we got to where we could build microscopes and we could see microorganisms. Now our next big question. Where do these microorganisms come from? So back in this time, back in the late 1700s, early 1800s, people thought that life was formed by spontaneous generation. Fancy way of saying, you just put stuff together and poof, magically life happens. Okay? Now we all think, that's just dumb, right? We know better than that, okay? Well, you got to think about who was in charge back then. The church was in charge, right? If you were a scientist and you went against the church, you were banished or put to death, right? So that was kind of the church's theory, was that, you know, with God's spark, life magically happens, okay? Well, the first guy that decided that was crazy, his name was Virchow. And Virchow started the theory of biogenesis, which means life comes from life, okay? Which is how we all know it works now, right? But nobody could prove that. They went through a ton of experiments, and I'm not going to make you memorize a bunch of stuff you don't need to know, but a couple of the experiments that come to mind, Francesco Reddy and John Needham, they did some experiments. One of them took raw meat and sat it out on the counter and said, look, a week later, there's stuff crawling on the meat. Look, life just magically formed. It was maggots. Okay? That's what he thought they magically formed. Where do we know the maggots came from? Flies landed on the meat, laid eggs, and the eggs developed into maggots. But that was the church's proof. See, life magically formed on the rotten meat. Okay? So then another guy came back and repeated that experiment but covered the meat with saran wrap and said, see, the fly can't touch it. No fly, no maggots. But he just got sent out of the country and they didn't believe him. So anyway, make a long story short, tons of experiments happened. The only experiment that truly disproved the theory of spontaneous generation was completed by Louis Pasteur. Okay, when I say that name, you all would have never said, oh yes, I know that Pasteur disproved spontaneous generation. You'd have told me that Pasteur did what? He invented pasteurization. So that's what we all know him for, but that's really not that great of an invention that he did, or great of a discovery. Okay? The fact that he was able to disprove spontaneous generation is much, much more important. The only reason Louis Pasteur invented pasteurization is because he liked to drink. And back in the day, wine and liquor would go bad. It would rot. Okay? It would get sour very fast. So he figured out if I heat my liquor a little bit, it'll kill some of the microorganisms and it'll stay good longer. So that's actually why he invented pasteurization. Okay? So anyway, but Louis Pasteur is most important for proving you have to have life to make life. Okay? His actual experimental apparatus is still in the Smithsonian. If you ever get to go to Washington, D.C., you can see it. We still have it. It was that important to the scientific field. Okay. A couple other guys. Jenner was the first one to vaccinate people, but he had absolutely no idea what he was doing. Okay, so I don't make a big deal about him. He discovered that women that milked cows would get something called cowpox, so they couldn't get smallpox. He didn't understand why, but he just decided what I'm going to do is give people cowpox vaccines, and then they won't get smallpox. Okay, he had no idea why, but he is credited for inventing the first vaccination. We're going to talk more about vaccinations later. These last two guys, you're going to see their names throughout the semester. The first one is Lister. Lister invented aseptic technique. So what does that mean? He was a surgeon, and back in the day, if you needed surgery, they would just move stuff off the table, pick up a knife, a scalpel, wipe it on their pants, lay you down, and cut you open, right? And we all know that just sounds horribly wrong, right? But they didn't understand that they needed to clean the microbes off of the scalpel and things like that. So what Lister did, he started using chemicals to clean his surgical instruments and to clean the wounds of his patients. And his patients started living. Other people were still dying. So they all thought, oh, this sounds pretty good. And the chemical he used was called phenol. We don't use that much anymore because it burns like heck. But we use stuff very similar to phenol. Okay? How many of you used Listerine when you left the house this morning? Okay. I asked that yesterday, and very few people raised their hand. That's very scary. You all need to do that, okay? It's good for your breath and the bacteria in your mouth. But if you use Listerine, it burns, right? It burns because it's got alcohol in it. Alcohol is very similar to phenol. It makes your breath smell better because it kills the bacteria. If your mouthwash, it's called Listerine. It's named after Lister. Okay? If your mouthwash does not burn, it just tastes minty, it's not doing anything. It just tastes good. Okay? It's supposed to burn. All right? Sound like your mom now. It's supposed to burn. That's a good thing. All right, last guy, Koch. Koch was the one that actually proved microorganisms cause disease. Before he did his experiments, we thought, oh, you've been bad, God is punishing you, and now you're sick. That's what we thought. We would bleed people, try to get the bad blood out. Okay, not a good idea. What Robert Koch did, he had some cows that were sick. He knew what they had. We know they had anthrax, but he didn't know. He just said, this cow's sick. He took some of the sick cow's blood, looked at it under the microscope, and said, look, I see stuff in his blood. I bet that stuff, the bacteria, is making him sick. So he took the sick cow's blood, injected it into a healthy cow, right? Healthy cow got sick and died. Then he took some of the second cow's blood, and he saw the same stuff in his blood. So he proved it was really the bacteria from cow 1 that made cow 2 sick. Very simple experiment, but very important. Okay? All right, so I know that was kind of just a brief overview, but I think it's important that you have an idea of what these major people did. Last couple things. One term. What does chemotherapy mean? Cancer, right? That's what most people think, right? You hear chemotherapy, that you think that means a treatment for cancer. It's really not what the word means. The word chemotherapy means using a chemical to treat an infection. So you need to start using the word properly. I've had a sinus infection, so I've been taking amoxicillin, right? It's the most generic antibiotic they'll give you. That is technically chemotherapy because I was taking a chemical to kill the bacteria that I had in my sinuses. Okay? We're going to talk about antibiotics, things like that, treatment. So you need to know the word chemotherapy is just generic treatment. Does anybody know what Alexander Fleming did? Anybody know that name? He was the first person to discover an antibiotic, the chemotherapy. Okay? He discovered penicillin. He did it by accident. He was working in the micro lab, had bacteria growing on Petri dishes. Came in the next day, I'm sure he said a few ugly words. There was mold all over his bacteria plates. So he went to throw them away. He had a moment of genius on his way to the garbage can. He looked at it and he said, I wonder why the bacteria aren't growing near the mold. I bet there's something in the mold that's killing the bacteria. So he took that mold and he purified the very first antibiotic out of that mold. And that's where penicillin comes from. It comes from a mold. Okay? Now, I don't know if any of you know Mr. Schultz. Any of you know who that is, right? He was our biology instructor here. He retired this spring. And the last thing he told me before he left this spring was that it was my job as the micro instructor to tell some of his stories. So I'm not going to, it won't take me 45 minutes to tell one of his stories like it used to take him. I can do it real quick, okay? But one of the coolest things that guy ever told me was that he was the first person in Mississippi to get a penicillin shot. Okay? And y'all may think, well, that doesn't sound very impressive. We've all had an antibiotic shot. Okay, we've all had a purified shot that was tested on, 50 lab rats before, and monkeys before it came to us. The very first penicillin shots, they just grew big vats of the mold, sucked the mold up in a syringe, and squirted it in somebody's butt. Okay? So you think about it that way, that's pretty impressive that he was one of those guinea pigs to get that test shot. So I just always thought that was kind of neat that he was the first one in Mississippi, the first group to get one of those penicillin shots. Okay? That's why, if you ever heard a lot of older people say they're allergic to penicillin, it's because of the way they used to make the penicillin. It used to just be basically mold, and it's not anymore. Now we don't even take penicillin, we take amoxicillin, which is a lab-made synthetic version of penicillin. Okay? All right, we'll talk more about antibiotics later. Okay? Not going to worry about these. We're going to cover each of these terms as we go through. Just put them in your PowerPoint so you would have them. Now, where I want to end today is showing you this last slide. Okay? At the end of every PowerPoint slideshow, you will see this. Okay? Of course, it will say do your own line assignments. But what this textbook study questions, multiple choice, this is not really something you have to do. These are the questions at the end of your chapter. Okay? The question and the answer are in your book. So it's not really an assignment I'm giving you. I'm just telling you, if you understand these at the end of your book, then you probably have a pretty good handle on the chapter. Does that make sense? So I just kind of went through to tell you which ones I thought were most important. Don't look at all of them. Some of them are crazy complicated. Okay? All right? Any questions?